all of you to Anjan Bios. I am Anjan Saranidar from TRB Academy, Department of Biology. Today we are going to discuss about double fertilization in Angiosperms. You see, this is the structure. This is a diagrammatic structure how fertilization takes place, and it's a microscopic view of a typical pistil, where this is the stigma, this is the style, and this part is ovary. Before going to study about fertilization, we should know some important point. We know this is the second phase or second part of sexual reproduction. In the first part, we have already discussed about pre-fertilization stages, where we have discussed microsporogenesis and megasporogenesis. During microsporogenesis, microspores are formed, that is, pollen grains are formed within the anther or microsporangium, while in megasporogenesis, megaspores are formed, then megagametogenesis takes place. After megagametogenesis, we have one 8-nucleate 7-cell embryo sac. Here is the 8-nucleate 7-cell embryo sac, this one. So then we have studied pollination. Pollination is nothing but the transfer of pollen grain from anther to the stigma. So this is the second phase where syngamy will occur. This process is called fertilization. Male gamete fertilize with the female gamete and produce a diploid zygote here in this process. For fertilization, a large number of pollen grains come to germinate over the stigma like this. You see, these are large number of pollen grains. These are landed on the stigma. After landing on the stigma, pollen pistil interaction takes place. Pollen pistil interaction means the stigma recognize its compatible nature of the pollen grain, whether it comes from the same species or different. This one recognized by this stigma, they secrete certain chemical, and after landing on the stigma, that is, after successful pollen pistil interaction, the pollen grain produce a tube-like structure like this. We know that before dispersal of this pollen grain, the pollen is surrounded by a two-layered cell wall. The outer one is called axin and inner one is called intine. The axin is made up of sporopollenin, and this axin in certain species they are very thin, and these species are known as germ pore. So pollen grain actually does not pass down the stigma, only its pollen tube does so. Whole pollen grain, it does not pass through the stigma, it produces a pollen tube. The intine along with its content, you know, the inner membrane of the pollen grain, which is called intine, along with its content emerges out from the germ pore as a short cytoplasmic outgrowth. This is called pollen tube. Each and every pollen grain produce one pollen tube, but the stigma and the style allows only one pollen to pass its pollen tube through the style. The pollen tube eats its way through the solid part of the style by secreting proteinase enzyme and some hydrolytic enzyme. This is the long style part. In some species the style is short, but generally it is a long one. So this pollen tube pass through this style, eating its content, and it produce or secrete pectinase and some hydrolytic enzymes. Then pollen tube travels intercellularly and chemotropically along the concentration gradient of calcium boron inositol sugar complex. Here a chemical complex is there. This chemical complex helps to pass the pollen tube through the style. In certain plants, pollen grain shed at two cell stage. Generally, within the pollen grain, it has two cells. One is called vegetative or tube nucleus. It is present on the tip of the pollen tube here, and another one is generative nucleus. The vegetative cell or the tube nucleus present at the growing tip of the pollen tube and ultimately degenerate. When this pollen tube reached here, then this tube nucleus degenerate. The generative nucleus divides mitotically and form two male gametes in case of the pollen grain which shed at two cell stage. But in certain plants, the pollen shed at three cell stage. Then pollen tube carry two male gametes from the beginning. Sometimes here generative nucleus divide to two and it form two male gametes and which carry this pollen tube from the beginning. The pollen tube generally grows further and finally reach to the micropylar end, and it reach one of the synergid. See, here is the embryo sac ready for the fertilization. This is the 8 nucleate embryo sac here. This is 2 synergid and 1 egg. Whole structure is called egg apparatus, and 3 cellulose end cells. These are known as antipodal cells, and 2 polar nuclei here. This is the 7 cell 8 nucleate embryo sac is ready for the fertilization. In the ovary, the growth of the pollen tube is directed by another tissue called obturator. The pollen tube enters the ovule either through micropyle. This is generally occur and it is known as porogamy. You see, this is the micropylar end, this is salada end, and the pollen tube enters through this micropyle, then it is known as porogamy. Or sometimes it may enter through the salada end. Here is a salada, then it is called saladogamy. And sometimes it may enter through the integument of the ovule, and this is called mesogamy. So the entry of pollen tube may be of 3 types. It may be porogamy when this pollen tube enters through this micropyle, and when it enters through the salada end, then it is called saladogamy, and when it enters through the integument, this is called mesogamy. In the ovule, the pollen tube is attracted by secretion of synergids. Usually the pollen tube enters the embryo sac by passing into one of the 2 synergids here. This impact destroys that synergid. The pollen tube also breaks open to release its content. One of the 2 male gametes one fuses with egg or oospheres, this perform generative fertilization. Two male gamete is here, you see. So one of the male gamete pass through one synergid after destroying the synergid, and it fertilizes with this egg or oosphere, and this is called generative fertilization. This generative fertilization is also known as syngamy. The second male gamete then slowly pass through it, and it fuses with this polar nuclei. This is known as vegetative fertilization, and it will form a triploid Thyssen number of chromosome, these cells 1, 2 and 3. This is called triple fusion. It becomes the primary endosperm nucleus or primary endosperm cell. Then we have two fertilization here. One is called generative fertilization, where one male gamete fertilizes with the egg and produce a diploid zygote. It will finally form the seed, or embryonal differentiation takes place from this zygote. Then the second male gamete, they fertilize with these two polar nuclei. It will form a triploid nucleus or triploid cell. This is called primary endosperm cell or primary endosperm nucleus. It will form the endosperm. As two male gametes fertilize twice, one is called generative fertilization, another is vegetative fertilization. So the fertilization of angiosperm is known as double fertilization. This is, you see why it is called double fertilization, because two male gametes fertilize twice. This is all about your double fertilization in angiosperms.